Our last video in this series is on the leaves, which is the last organ of the plant that we are studying. First of all, let's look at some external features of a leaf that you need to know. And you'll probably have a diagram on this on your quiz. The blade is the flat portion of the leaf. It's usually what a person refers to. Actually, they usually refer to the leaf as being the blade and the petiole. But the blade is that flat, usually green portion. Then the petiole is the little stalk at the end of the leaf that attaches that leaf to the stem. So a leaf really is the whole structure from where the petiole is attached to the very tip of the blade. That's the leaf. Around the edge of the leaf, that is called the margin. And then you should be familiar with veins. Technically, a vein is a fibrovascular bundle, and they are the vascular tissue, the pipelines that are carrying the food and the dissolved water. And the main vein, as you can see in this particular plant, when there's only one main vein, they actually refer to that vein as a midrib. And then the place where the petiole, where the leaf is attached to the stem, that particular place on the stem is known as a node. And then sometimes you don't see these, sometimes you do. It's what's called a stipule, and that is where the petiole attaches. It looks like usually little green, small little tiny leaves, or maybe brown in color, or they may actually die and fall off, because once the leaf emerges from the bud, the stipule is no longer needed, because the stipule was that small leaf-like or scale-like structure that covered that little tiny baby leaf as it was developing inside the bud. And so that's called a stipule. There is two different kinds of venation patterns, and this is when you look at the veins on the particular leaves. There's parallel venation, which if you know what parallel lines are, that should be pretty easy to remember, and then netted. So parallel, netted, if you think branched, that will get you netted venation. And then under netted, there's actually two types, which you can see pictured up there, palmate and pinnate. So parallel venation, the veins run side by side, sort of like the railroad tracks. And that's parallel lines do the same. Now they will merge at the top of the tip. So it's a series of veins which originate the stem and proceeds to the tip of the leaf. It occurs in your monocots, so it's going to be in the corn that you are growing. It's in grasses, irises, orchids. And then with netted venation, which is branching, there's two kinds that you can see. You can see where there's more than one main vein, which comes to a single point, such as the ivy, the maple, and the geraniums. Or you can have that midrib, that one main vein, and then you have branching off of it, such as oak trees, apple trees, and African violets. Now, leaves are classified as being either simple or compound. A simple leaf is what you're going to find on most of your trees. There's one blade on each petiole, and you can see the petiole labeled, and you can see the blade labeled. If it's a compound, it's actually going to have several blades on a petiole, not just one. This is the common leaf, the frond on a fern. So each of those little tiny small blades on the compound is a leaflet, not a leaf. And it has a quite long petiole. Sessile leaves are leaves that do not have petioles. So the way that they are attached to the stem is they wrap around it. Excuse me. So sessile leaves, or certain other monocots have sessile leaves like grass. They're attached to the stem by way of a sheath. That's what wraps around the stem. And here's a good example showing you. Now the mosaic. You need to know that the leaf mosaic is how the leaves are arranged on a stem. And there's 3 different types. Be able to list them and know the difference. There's alternate leaf mosaic. So there's alternate leaf mosaic, opposite leaf mosaic, and whorled leaf mosaic. And let's look at the difference and what they look like. Right here you see the pattern. When you think of alternating, notice how that the petioles are attached on opposite sides of the stem, but not at the same place. So that is called an alternate mosaic. And the key that you need to know for the quiz is that there's 1 petiole per node. And one's on the right side, then the next one's on the left side. That's why it's called alternate. Opposite leaf mosaic, what you need to know is that 2 petioles are per node. So you see one on the right, then at the same place on the left-hand side of the stem, you see the other leaf. And that's called opposite because they're on opposite sides at the same place. And the whorled is what you see a lot in the tropical types of plants, and it's when you have more than 2, so you have 3 or more leaves growing at a single petiole. A single node. Here's a drawing showing you the opposite, alternate, and whorled. Now leaf shapes, we're going to go over these. You don't need to know these for a quiz. But there are different leaf shapes, and this is all, again, still the exterior part. And we're going to look at each. Here's a good example of linear. Linear means line, so they're long and narrow. Lobed, you can see how they're lobe shaped. They don't come to points. Then there is the heart-shaped ones, or kidney-shaped. They're called cordate. Deltoid is like a triangle, so it doesn't dip in like the heart, it goes straight across. And deltoid is a triangle leaf shape. And then, of course, circular is round. There's also leaf margins, and again, you don't need to know this for the quiz. We're going to look at each. Entire is where it's nice and smooth all the way around. When you have some teeth on it, toothed margins, it's called serrated, and that's like a serrated knife, which has all those teeth to cut. Undulate is when they're wavy instead of pointy, not like a tooth, but they're wavy margins. Dentate is real tiny points, or like stickers coming out, and those are teeth pointed out. Now, the rest of this you do need to know, the internal structures of a leaf, and you will have a diagram on this as well. And we're going to look at the different structures then that are inside of the leaf. Most leaves are the major site of photosynthesis, so that's the organ, that's the function of the organ of the leaf, is to produce sugar, glucose. And the top and the bottom layers of the leaf are made of epidermal cells, 1 cell layer in thickness. So remember, the roots, the stems, and the leaves are all covered by an epidermis, unless you have a woody stem, then it's covered by the bark, which is cork. So we're going to look at the epidermis here. It's always 1 cell layer, so see where the lines are pointing, that top layer, and then the bottom layer is the epidermis of the leaf. So it's the top and bottom layer. It's only 1 cell layer in thickness. It doesn't have any chlorophyll, so that's not where it gets its green color. It's actually clear, and you can pull it off. It serves as protection, and sometimes remember that there could be an extra, not cellular level, but it's a coating on top of it, which is called a cuticle. And they're usually transparent. Why? Well, think about the green parts underneath that need sunlight. So the sunlight has to be able to get through. So the cuticle we've already talked about, but again, you need to know that's the waxy substance that's made by some epidermal cells, which is added protection for the leaf. The lower epidermis will usually have several, and it depends on what type of leaf, as you'll see in a minute, little tiny openings. Those little tiny openings are called a stoma singular or stomata plural. They're leaf pores, leaf holes. And they're there because the plant has to get rid of the extra oxygen, but it also has to take in carbon dioxide. So these stomata are very important for the exchange of gases to take place between the atmosphere and the leaf cells inside. It also has another function, which is to release water, which is called transpiration. So the main purpose is to allow air to move in and out of the leaves. Most leaves, here's a good example showing you the stomata. They're surrounded by two structures that are called guard cells, and that is what is opening and closing them. When the stomata are open, the carbon dioxide and oxygen and water vapor can pass through, in or out, whichever the case may be. The lower epidermis, and you don't need to know these statistics, but this is showing you an example of how many of these stomata there are. On an apple tree leaf, just one leaf, there's about 47,000 stomata per square inch. And on an oak tree, it's 100,000 per square inch. So they're easy to find under the microscope, which we'll be looking at them. And then, as I said, there has to be something that opens and closes them, and that's the guard cells. And they are crescent-shaped. They surround the stomata, and their function is to open and close the stomata. Now, they do not have muscle. So how would guard cells open and close the stomata without having muscle tissue to do that? Well, it's by turgor pressure. So here on the top you can see the stoma, which is the opening, and the two guard cells on the side. At the bottom, that's a closed stomata. So you see the guard cells, and then the line between the guard cells is the closed stomata. Here showing you again some open stomata, and then one that is closed on the guard cells surrounding them. And here's what I was talking about with turgor pressure. So you need to know it's turgor pressure that is opening and closing them. And in order to open, there has to be the water entering the cells, so that would be an increase in the turgor pressure. And then for them to close, the water has to exit, which would cause them to decrease. Here's some more showing you some guard cells. Here's I like this picture because this is a real picture, and you can see two guard cells with open stomata. Now, some other things that you need to know internally are the right under usually the upper and lower epidermis, and that's what's called the mesophylls. And it's actually a parenchyma, so it's a ground tissue. And you need to know in which mesophyll is most of the process of photosynthesis occurring. And it's actually in the first one listed there, the Palisade Mesophyll. And there's also Spongy Mesophyll. So, see the upper epidermis? See those long tube-looking, like columns stacked side by side? That's the Palisade. Then if you look at the bottom, the Spongy Mesophyll is got a lot of spaces in it. That's why it's called spongy. And then you have the lower epidermis. Now, this particular leaf has two layers of Palisade Mesophyll and one of spongy. So below the upper epidermis are rows of tightly packed cells that are called Palisade Mesophyll cells. And know that this is where most of the photosynthesis occurs. Palisade Mesophyll. They have a lot of chloroplasts. So it's Palisade, located toward the upper side of the leaf consists of elongated column-like cells. There may be more than one layer, as we saw on that one, and there's an abundance of chloroplasts. And it's interesting, if you watch a living cell, they actually will be just under the cell membrane, and they circle around and around and around. And the reason they're going around and around and around is because at the top they're getting sunlight, and at the bottom of the cell they're going to be getting the water and the carbon dioxide that they need through the spaces in the spongy mesophyll. So spongy mesophyll is located usually in the lower, although there's some leaves that would have a palisade, then a spongy, and then another palisade, but that's not the usual. It consists of large, irregularly shaped cells, and they have lots of air spaces in them, and those air spaces are actually forming passages in the leaf that will allow the air to come in so that the cells are exposed and can give oxygen out and take in carbon dioxide, and also where the water coming up by way of the xylem can get in those spaces and give each cell the water that they need. Veins, you need to know, are actually fibrovascular bundles, but they're commonly referred to as a vein in the leaf. And so they're running through the mesophyll, and of course, we've already learned this, they contain the vascular tissues, xylem and phloem, and they're surrounded by supporting tissues, as you can see, that thing that to me that looks like a monkey face. So they contain thick-walled, strengthening collicumin tissues. You don't need to know that last statement, but that is what surrounds them and holds them together. Here's a nice diagram showing you the things that we've talked about. Your diagram on the quiz will not have color, obviously. Here's one from an electron microscope, and see if you can easily spot all the parts that we were talking about. And we're going to go over these in class, but see at home if you can spot the different parts. Here's one that we have as well in our slides. Notice in the very center, that big area that's circular, that is a midrib. That's a big fibrovascular bundle. Some more examples of you to find the stomata, the guard cells, the epidermis, the fibrovascular bundles, the mesophylls. Here's one that has them labeled for you. Here's another diagram. Now, you need to know how gases are exchanged in a plant. And it's different parts, different ways. And when you're talking about the root of the plant, we'll get into that later. That's actually number 3, I believe. So the first is, you've got two processes going on in the green cells, the ones that have chloroplasts. And you have not only photosynthesis, but remember, every cell has to have cellular respiration. So you have oxygen left over from the photosynthesis, which is going to be used for the cellular respiration, which gives off carbon dioxide, which is going to be used for the photosynthesis. So what you need to list on the quiz, when I ask for you to list the ways gases are exchanged in a plant, the first one you would put the cycle of photosynthesis and cellular respiration. So make sure you put it that way. The cycle of photosynthesis and cellular respiration. In the leaves, you also have the stomata. So that's gas exchange. So you got two things going on for gas exchange in the green leaves. In the stems, God put little tiny holes as well, but they're not surrounded by guard cells. They constantly stay open, so they're not closed, and these are called lenticels. Lenticels. And so they are the small openings in the woody parts of plants. So this would not be a herbaceous stem, but a woody stem. And then in the epidermis, this is for the roots. The roots they're underground. How do they get oxygen? From air pockets down there. And so when the air pockets are close to the epidermal cells, they will allow an exchange of gases by diffusion. And this is where, remember when we studied about the worms, this is one reason why they're important in aeration. Because when those little creatures are digging, they're putting nice little holes and tunnels down there that air can get down deeper into the soil, which helps the roots. Now there are some special plants that have special, and you don't need to know this for the quiz or a test, but they actually have their own special way of gas exchange because they live in water, and so their roots would be actually under the water. Now the last thing is, what do plants do with all this food that they're making? Fortunately, remember, they are the producers, so they're making more food than they need, which is a good thing because this is how we live. We live off of the producers. So glucose, remember, contains in that stored form the energy that's been captured from the sun. And that's in the glucose. That's why it's food. It's got that potential energy. So cells carry on photosynthesis, and they're making a lot more sugar. So here's where that sugar is going to. In some, the glucose molecules are not used for energy, and they're hooked together to make cellulose. Now remember, some they're using on their own to use in cellular respiration. But some are hooked to make cellulose, and this is the good part for us, some are stored as starch or oil or lipids. And that's what we're eating, like potatoes, wheat, bananas, corn. They have a lot of glucose in them. And of course, whenever you use the oils or when you eat a food that has oil in it.
A typical leaf is made of several different layers of cells. On the upper and lower surfaces of a leaf is a thin layer of cells called the epidermis, which covers and protects the leaf. A waxy cuticle coats the epidermis of some leaves. Most leaves have small openings in the epidermis called stomata. Each stomata is surrounded by two guard cells that open and close it. Just below the upper epidermis is the palisade layer. Between the palisade layer and the lower epidermis is the spongy layer. In a leaf, veins containing the vascular tissues xylem and phloem are found in the spongy layer.